Hello everyone. I would like to thank the conference committee for selecting my abstract and for broadcasting this event despite current circumstances. Throughout this presentation, I will show you the influences of latency reversing agents on HIV infection in human primary macrophages.
I wish, but I have nothing to disclose. As you may know, latently infected cells represent one of the main barriers to HIV-1 cure in patients under antiretroviral therapies. This mechanism has been identified in CD4 cells where HIV-1 DNA, here shown in red on this cartoon, is integrated but does not produce viral particles, allowing immune and therapeutic escape. Thus, by reversing latency, a phase called shock, the ensuing viral production associated to antiretroviral therapy to limit viral spread will allow clearance of these cells by the host immune system or by viral toxicity. This so-called shock and kill strategy would thus be able to purge latently infected cells and eradicate HIV. Extensive research has been made using small molecules called latency reversing agents or LRAs in order to achieve the shock phase. On this chart are represented the three main classes of LRAs used in scientific studies: histone deacetylase inhibitors, protein kinase C activators, and bromodomain inhibitor. No matter of the mechanism, these agents were all shown to increase HIV-1 viral transcription. Indeed, many studies show that LRAs can reactivate HIV-1 production in latently infected cells ex vivo and in vivo without causing global T-cell activation or toxicity. However, these agents are not cell-specific, and their effect on non-lymphoid cells are mostly unknown. Among those cells, macrophages represent one important target during HIV-1 infection and are latently infected in vivo. Yet, no data are available on LRA's impact on macrophages. Therefore, our team aimed at evaluating human macrophages' susceptibility to HIV-1 infection and modulation of viral production by LRA's stimulation. We included one of the best characterized and potent agents for each LRA's classes previously mentioned, which are romidepsin for histone deacetylase inhibitors, bryostatin-1 for protein kinase C activators, and JQ1 for bromodomain inhibitors. These agents were tested alone or in dual combination and used at concentrations achieving the best potency to reverse latency in CD4 T cells based on previous report. First of all, we wanted to assess the toxicity associated to LRA's treatment in macrophages. In our experiment, we used monocyte-derived macrophages from healthy donors. The experimental procedure is displayed at the bottom of the slides. Here, 24-hour stimulated macrophages were analyzed by flow cytometry for cell viability by fluorophore exclusion. As you can see on this graph, except for TNF and interferon gamma conditions used as a positive control, none of these LRAs used alone or in combination impacted cell viability when compared to the non-treated cells. Likewise, apoptosis detected by flow cytometry was not triggered by LRA's treatment, as you can see on the right panel. We thus concluded that LRAs are not toxic for macrophages after 24 hours stimulation. Since some LRAs were shown to affect HIV-1 receptor expression in CD4 T cells, we assessed this phenomenon in macrophages. Macrophages stimulated for 24 hours with LRAs were quantified for their HIV-1 cell surface receptors, CD4 and CCR5, by flow cytometry. As presented in the left graph, romidepsin treatments led to an important decrease in CCR5 positive cells, while other LRA had no effect. Not shown here, we also observed a tenfold decrease in CCR5 RNA expression with romidepsin treatments. Meanwhile, bryostatin treatments induced a twofold decrease in CD4 positive cells, shown in the right panel, while romidepsin, used in combination with bryostatin-1, appears to rescue CD4 expression to normal levels. Since receptor expression could influence viral entry, we next investigated the consequences of LRA stimulation on HIV-1 infection. To do so, macrophages were stimulated for 8 hours, then infected for 16 hours with an R5 reporter virus. Cells were then cultivated for 3 days in the presence of efavirenz to ensure a single round of infection and to allow proper reporter protein expression and quantification by flow cytometry. As you can see, romidepsin treatments induced a twofold decrease in the percentage of infected cells, while bryostatin 1 induced a nearly complete resistance to HIV-1 infection. In order to assess whether receptor expression is involved in this phenomenon, we used a VSVG pseudotype reporter virus to bypass CD4 and CCL5 usage. As depicted on the right panel, romidepsin induced a similar reduction of infection with this pseudotype virus. However, bryostatin 1 caused only a modest 25% inhibition of infection. Thus, we concluded that HIV-1 infection decrease is receptor independent for romidepsin, while it is partially dependent for bryostatin 1. Finally, we assessed LRA's effect on viral production. Here, macrophages were first infected for 24 hours and then cultivated for 11 days with efavirenz to promote possible latency establishment. Cells were stimulated with LRAs for 24 hours, supernatant were collected, and the P24 protein was quantified by ELISA. As depicted on this graph, none of the LRAs tested were able to increase HIV-1 viral production. This could imply either that no latently infected cells were present in our culture condition, or that the LRAs used were not able to reactivate these cells. On the contrary, bryostatin 1 stimulation induced surprisingly a massive drop in P24 extracellular detection. To assess if a virion release impairment could explain this reduction in viral production, we then quantified the intracellular or cell-associated P24 production. As you can see, the same trend as our extracellular result was observed using bryostatin 1 with a twofold decrease in cell-associated P24. This result tends to imply that viral release blockade was not the main mechanism involved in viral production impairment induced by bryostatin. Since viral production silencing in infected cells is a hallmark of latency, one might think that bryostatin 1 could promote latency establishment in macrophages. To test this hypothesis, we used the same protocol as in the previous slide and quantified GAG and TAT splice messenger RNA expression by PCR. Our results show that none of the treatments could modulate either one of these genes. This led us to conclude that bryostatin 1 does not exert a transcriptional block on HIV-1 expression. To sum up the study, we showed that LRAs are well tolerated and that romidepsin and bryostatin 1 infection decrease are mostly and partially receptors independent, respectively. Although these results are quite new for in vitro human primary macrophages, they are similar to previous reports on CD4 T cells and cell lines. Moreover, with minor exceptions, no combinatory effects were observed. No evidence of latency reversal was seen during our viral production experiments, and we are not trying to detect latency establishment in our MDM model. But most importantly, we showed for the first time to our knowledge that bryostatin 1, a latency reversing agent, in fact decreases HIV-1 viral production in macrophages. While this was not linked to transcriptional block, this agent could still be promoting latency in macrophages by post-transcriptional silencing, which is currently under investigation. Our work shows that different outcomes of some LRA stimulation among cell types exist and could be potentially harmful. In the light of our results, it seemed crucial to investigate LRAs or other cure strategies side effects on a wider cellular spectrum. I would like to thank all my team, especially Marc-Olivier Tormel, for his work on this project, the funding partners, blood donors and nurses, which make our experiments possible. And thanks again to the THR committee for the organization of this event.